Hello and welcome to Genetics Day 4. Today's aim is how does DNA transcribe its information outside the nucleus? So today we're really going to be talking about how DNA is used to build proteins and how DNA actually functions as the instructions for life. So first, a little basic review. We want to remember all teachers go crazy and build the complementary DNA strands to the following template strands. So go ahead and pause this video and try to build those complementary DNA strands. Did you pause the video? I hope you did. So we have AT, AT, GC, TA, CG, TA, GC, CG, AT. A always bonds with T, G always bonds with C, although we're about to add another level of complication to that, but not yet. So the second one, CG, GC, TA, AT, GC, TA, CG, AT. All right. So part A, DNA structure notes. So DNA has the information, there we go. DNA has the information to build from DNA to proteins to cells to bodies. So DNA has the information to build proteins. How do the proteins do all the work? Genes, so the DNA has the instructions to build the proteins build the cells, and the cells build the bodies. DNA gets all the glory, but protein does all the work. Genes run living organisms. Proteins run living organisms. Forgive me, the PowerPoint is a little out of order with your notes. Enzymes control all chemical reactions in living organisms, and enzymes, as you know, are proteins. So, okay, let's go back. The first blank, DNA has the information to build proteins. How do proteins do all the work? Proteins run living organisms. Second blank. Third blank, enzymes control all chemical reactions in living organisms. Fourth blank, structure. All organisms are built out of proteins. Wowza. Pretty cool stuff. Proteins. They're even more important than you may have previously thought. Cell organization. DNA is mostly found in the nucleus, with the exception that the mitochondria and the chloroplasts have their own DNA. Genes, and we want to keep it there, and it's protected, locked in the vault, right? So within the cytoplasm, we have the nucleus and their nuclear membrane. Within the nucleus, we keep the DNA locked inside the nucleolus because those instructions are very, very, very important and we don't want them to get damaged. So genes are the instructions for making proteins, and we want to keep it in there, protected, found in the nucleus, blank number 1. Genes are the instructions for making proteins, blank number 2. Want to keep it in there, protected, locked in a vault. Proteins are chains of amino acids, which are the monomers of proteins, hopefully we remember, made by a protein factory in the cytoplasm. And the protein factory, as I hope you remember from Unit 2, are ribosomes, which are found in the cytoplasm and on the surface of the rough endoplasmic reticulum, which is called rough ER because it's covered in those little bumpy ribosomes. Those are chains of amino acids. Those are proteins. And the ribosomes are the protein factories that actually build the proteins. Thanks, ribosomes. So passing on DNA information, need to get DNA gene information from the nucleus to the cytoplasm. How do we do it? Because the information from the DNA needs to make it to the ribosomes so they can actually build these proteins. We need a copy of the DNA, which uses messenger RNA. And here's where we add a little complication to our ATGC rule when we talk about RNA. So this messenger RNA is very important and it is involved in the process of transcription. So the DNA from the nucleus is transported out of the nucleus as mRNA, which makes its way to the ribosome, where it then is used as the instructions to build proteins. And I just said transported, but we're talking actually about transcription. So what is transcription? Transcription means using DNA to make mRNA. So this is where the complementary rule is very important. The DNA strand is the template used to make mRNA. So we have one strand of DNA, and then we're building a complementary strand of mRNA. But in mRNA, there is no T. Instead, there is only uracil. So adenine bonds with U, uracil. So no thymine, only uracil. Uracil is only found in RNA molecules. So no T bases in RNA, so U matches with A instead of T, A, U, and RNA polymerase, just like we talked about DNA polymerase, is the enzyme used to make an mRNA strand. And then on your paper, you have some nice images of replication, how we make a complementary strand of DNA, which is DNA replication, where we're using that ATGC rule, and then RNA, which is single-stranded, which is made from that same template, but then becomes just a single strand that is carried out of the nucleus to the ribosome as instructions for the construction of proteins. So matching bases of DNA and RNA. The double-stranded DNA unzip, and then the RNA bases match to the DNA bases on one of the DNA strands, and then RNA polymerase zips it back up to create, or rather the RNA polymerase is what pulls in the bases to actually make the strand of messenger RNA. So the RNA polymerase, just like the DNA polymerase, matches each base according to the base pairing rule, which is slightly modified here because adenine matches with uracil instead of thymine, which also means thymine then is going to match with adenine still. So U instead of T is matched to A. So now we're going to read part D, making sense of transcription, because I bet you have a few questions. And you can see here it follows the same base pairing rule we've been talking about, except T is replaced with U. So anytime, and you can see there the ribosome then reads that strand of messenger RNA and produces proteins. And we use something called the universal codon chart to actually figure out what proteins are constructed based on these instructions. So the nucleus that keeps the DNA safe and protected. The DNA is still kept in there. It's not ever removed from the nucleus, but the messenger RNA is able to travel outside of the nucleus to the ribosomes with this slightly altered construction so that proteins are built. And then those proteins determine our traits. All right, genes and proteins. Now that it has been shown that DNA is what makes up the genetic material, it is time to look more closely at genes. What is a gene? A gene is a specific segment along a DNA molecule made up of linear sequences of nucleotide subunits. It is, in effect, a package of information that tells a cell how to make proteins. Likewise, proteins are polymers made up of a linear sequence of amino acid subunits. There are 20 different types of amino acids. The order in which they assemble determines the type of protein that they make. We will learn more about this tomorrow. This sequence determines the shape of a protein molecule. It is the shape of the protein that allows the molecule to do its work in the cell. Cells have to use the linear sequence of subunits in DNA to build amino acids for protein. Yet, in all cells except for bacteria, DNA is stored in the nucleus, and protein synthesis occurs outside the nuclear membrane at the ribosomes. These small organelles are distributed throughout the cytoplasm. How does the genetic information contained in DNA within the nucleus get out to the ribosomes? A third type of molecule called ribonucleic acid, or RNA, works as a helper to transmit the information. That is, the genetic information flows from the DNA to the RNA to the protein. So, what is a gene? A gene is a specific segment along a DNA molecule made up of linear sequences of nucleotide subunits. It is a package of information that tells a cell how to make proteins. What determines the shape of a protein molecule? The protein molecule shape is determined by the RNA sequence that provides instructions to the ribosome to build the protein. The order in which the amino acids assemble determines the type of protein that is made, and subsequently, its shape. So those are questions 1 and 2 under genes and proteins. From DNA to RNA. Each gene is a portion of a chromosome, in effect, a portion of the DNA chain. An RNA molecule called messenger RNA does the job of moving the information in the linear base sequence out to the ribosomes. DNA is copied into RNA in a process called transcription, which is similar to DNA replication. First, the DNA double helix, and again, it's extremely similar to DNA replication. The difference here is that there's one different base that is used, uracil, and a different enzyme, RNA polymerase, but it is building a complementary strand of RNA, just like when DNA is replicated, it's building a complementary strand of DNA. So it's a very similar process, except in the end, the outcome isn't one double helix DNA molecule, but rather a single-stranded RNA molecule that is going to leave the nucleus and be used as instructions to set up amino acids in order to build proteins. First, the DNA double helix opens up where a particular gene is located. Special enzymes begin to match up RNA subunits with the correct DNA subunits. The new RNA molecule has the same base sequence as one strand of the original DNA, except in RNA, the base uracil substitutes for the DNA base thymine. This RNA molecule then goes out of the nucleus through pores in the nuclear membrane to ribosomes in the cytoplasm. How does genetic information contained in the nucleus get to the ribosomes in the cytoplasm? By being turned into a strand of messenger RNA. So RNA polymerase is involved in the process of transcription. So the short answer to number 3, how does genetic information contained in the nucleus get to the ribosomes in the cytoplasm? Through transcription. RNA polymerase is used to build a complementary strand of RNA, which matches the DNA in the nucleus and carries that information through pores in the nuclear membrane out to the ribosomes to be used as instructions for the construction of proteins. It's a mouthful. This is probably the most complicated thing we will study all year. Ooh, hold on. And we can see the double-stranded DNA is transcribed into the single-stranded RNA. We can see they're very similar in structure. The difference is RNA is single-stranded and DNA is double-stranded, that double helix structure, and the base thymine is replaced with uracil. Each gene is a portion of a chromosome, in effect, a portion of the DNA chain. An RNA molecule called messenger RNA does the job of moving the information in the linear base sequence out to the ribosomes. DNA is copied into RNA in a process called transcription, which is similar to DNA replication. First, the DNA double helix opens up where a particular... Did I skip this part? I swear I already read this information. Forgive me. So I think I already read that information. What happens during the process of transcription? RNA polymerase is used to build a complementary strand of messenger RNA that is carried out of the nucleus to the ribosomes. That is transcription. So the DNA is transcribed into messenger RNA, which is then carried to the ribosome and uses the instructions for putting the amino acids in the correct order to build proteins. Now we're going to write in the complementary mRNA bases for the DNA strand below. So the DNA, A, U, G, C, G, U, G, T, excuse me, G, U, A, U. Beautiful. So again, there is adenine in DNA, but even I make mistakes sometimes. There is no thymine. So when you see A, you're pairing it up with U. When you see T, you're still pairing it up with A. And these 3 string chunks of nucleotide bases are called codons. And we will take a look at the universal codon chart in a lab in the future. This is an example of the universal codon chart. And you can see there's a whole lot of U's in there, and those little abbreviations, those are the amino acids that these codons code for. All right, so that is all that I have for you today. Sorry, that was a little disjointed. Today's aim was, how does DNA transcribe its information outside the nucleus? What I'll go ahead and do is I'll upload a copy of this worksheet, these notes with all the answers on it alongside this video. So in case you were a little confused, you'll also have that to go along. But have a super fantastic day, guys. I'll see you all on Zoom on Thursday. Bye.